Hello and welcome to Biology Unit 2 Replication. So with all this talk and time spent on the topic of DNA, it only makes sense that we talk a little bit about what happens when you need to make more DNA. Because again, all the cells in your body have DNA, but your body often needs to make new cells. But if those cells don't have their own DNA, they'll have no instructions, no blueprint for what their purpose in life should be. And the topic of this video is when a cell divides, how does it divide? And how does it divide in such a way that its genetic code gets properly copied into the new cell? Well, how it does that is through DNA replication. Before a cell divides, it duplicates its DNA in a copying process called replication. This ensures that when a cell divides, like for example, when you get hurt and your body needs to heal that injury or that cut, and it does so by making a scab. Well, it does that by dividing cells to make new cells. But those cells need a complete set of DNA, and that's what replication tries to guarantee. And replication can begin at multiple places along the DNA molecule. There's many different places it can start. There's no one for sure, always has to stop and start here, proceeding in both directions until each chromosome is completely copied. So up here you see a complete chromosome being unzipped and two new strands coming up to meet it to make two new sets of chromosomes, two new sets of genes. But to make sure those two sets make sense is the DNA polymerase. And the DNA polymerase goes along these separated strands and follows Chargaff's rule, Professor Chargaff. You know that G's and C's have to match up. Anytime you see a T, you got to add an A. Anytime you have an A, you got to add a T. G's and C's, A's and T's. The order doesn't matter, you just can't mix up A's and T's with G's and C's. And as it does that, it creates a new strand so that eventually you get two new sets of genetic code. So to summarize, you start with an old strand, it gets unzipped. Then the polymerase adds new bases, A's, T's, G's, and C's, along both sides of the original strands, and then new strands are attached to those bases until you get new genetic codes mixed with the old and the new, but two new individual genetic strings, chromosomes that do not need to be reattached. And that's all there is to know for the moment on replication. For transcription and translation and a couple other topics, you're going to have a guest speaker, the Amoeba Sisters. But just as if I was speaking, please, if you have any questions, feel free to ask. Thank you.